Hello students, so we are doing the lesson plant growth and developments. Hope all of you are fine. So dear children, in our previous video we had done the enzyme cytokinin and gibberellin. So in this video we are going to discuss about ethylene and abscisic acid. Ethylene, it is a gaseous hormone which stimulates transverse or isodiametric growth but retard the longitudinal one. Businessmen dealing with storing and shipping of the fruit had known quite early that rotten or ripe fruit could trigger early ripening of other fruits present nearby. Cousins in 1910 found that ripe oranges produce a volatile substance that hastened ripening of unripe bananas nearby. With the help of gas chromatography, the GAN in 1934 found that the ripening causing volatile substance was ethylene. So ethylene was recognized as a plant hormone by the Crocker in 1935. Ethylene is produced in the plant from the amino acid methionine. It is formed in almost all plant parts, roots, leaves, flowers, fruits, seeds. Okay, maximum synthesis occur during the climacteric ripening of the fruit and tissue undergoing senescence. Excess of oxygen also induce ethylene synthesis. Many effects of excess oxygen are actually the effect produced by the ethylene. Now functions of the ethylene. Growth, ethylene inhibits longitudinal growth but stimulate transverse or horizontal growth and swelling of the axis. Gravity, it decreases the sensitivity to the gravity. Root become geotropic, while the stem turn positively geotropic. Leaves and the flower undergo drooping. The phenomenon is called epinasty. The seedling develop tight epicotyle hook. Senescence, it hastens the senescence of the leaf and the flower. Abscission of various part of the leaves, flower, fruits is stimulated by the ethylene, which induce the formation of hydrolase. Now, apical dominance, ethylene promote apical dominance and prolong the dormancy of the lateral buds. Breaking dormancy, it breaks the dormancy of the bird seeds and stored storage organs. Abscisic acid, it seems that formation of abscisic acid in the leaves under the condition of water stress is mediated through the ethylene. Growth of rice seedling, ethylene promote rapid elongation of leaf base and internode in deep water rice plant. As a result, leaves remain above the water. Root initiation, in low concentration ethylene help in root initiation or growth of the lateral roots and root hair. This increases the absorption surface of the plant root. Now fruit ripening, it aids in the ripening of the climatic fruits and dehiscence of dry fruits. Climacteric fruits are fleshy fruits which show a sudden sharp rise of respiration rate at the time of ripening. Respiratory climacteric, they are usually transported in green or unripe stage. Ethylene is used to induce artificial ripening of these fruits like mangoes, apple or banana. Flowering, it stimulates the flowering in the pineapple. Okay, the ethylene can stimulate flowering in the pineapple and related plant as well as mango, though in other cases the gaseous hormone cause fading of the flower. This help in the synchronizing sex of the sets of the fruit. Okay, sex expression, like auxin and cytokinin, ethylene has feminizing effect on the sex expression. Genetically, male plants of the can be induced to produce female flower in presence of ethylene. The number of female flower and hence fruit is enhanced in the monoecious plant like cucumber. Use of ethylene. Ethylene regulate a number of physiological processes, therefore it is widely used plant growth regulator in agriculture. The common compound used for obtaining ethylene is ethophenone, okay, or it is ethyl, which is 2 chloroethyl phosphonic acid. In aqueous solution, ethophenone is readily absorbed and transported to various parts. It releases ethylene slowly. The various commercial use of ethylene are fruit ripening, kerosene lamps, and hay were previously used for stimulating color development and ripening of the some fleshy fruits like banana, mango, apple, tomato. The effect is due to ethylene. Ethylene lamp are now specifically used for this purpose. Feminizing effect. External supply of very small quantity of ethylene increase the number of female flowers and hence fruit in the cucumber. Sprouting of the storage organs, rhizomes, corms, tubers, seeds, okay, for example, peanut and other storage organs can be made to sprout early by exposing them to the ethylene. Thinning excess flower and young fruit are thinned with the help of ethylene, for example, cotton, cherry, walnut. It allows better growth of remaining fruits. Next we have is abscisic acid. Abscisic acid, it is also called stress hormone because the production of hormone is stimulated by drought, waterlogging, and other adverse environmental condition. Abscisic acid is known as dormin as it induces dormancy in the buds, underground stems, and the seeds. Its another name are abscissine 2 and inhibitor B. Abscisic acid is a mild acetic dextrorotatory sesquiterpene growth hormone which functions as a general growth inhibitor by counteracting other hormones, that is auxin, gibberellin, cytokinins, or reaction mediated by them. The hormone was first isolated by Adicott, okay, in 1963 from the cotton balls. It is produced in many parts of the plant but more abundantly inside the chloroplast of the green cells. The hormone is formed from the mevalonic acid or xanthophyll. It is transported to all parts of the plant through diffusion as well as transport channel, that is phloem or xylem. Abscisic acid is helpful in the bud dormancy. Abscisic acid induces dormancy of the buds towards the approach of the winter. Then it is mainly caused, that is seed dormancy is mainly caused by the abscisic acid. Dormancy allow the seeds to tolerate desiccation and extreme of the temperature better. The bud as well as the seed sprouts only when abscisic acid is overcome by gibberellin because of its action in inducing dormancy. Abscisic acid or ABA is known as dormin. That is stoppage of the cambium activity is also induced by abscisic acid. Formation of abscisic acid stop mitosis in the vascular cambium towards the approach of the winter. Abscisic acid promote the abscission or aging of the flowers and the fruits. Its excessive presence stops the protein and RNA synthesis in the leaves and hence stimulate their aging or senescence, that is leaf fall, is actually promoted by the ethylene. Now transpiration during desiccation and other stress, abscisic acid is rapidly synthesized. The inhibitor cause closure of the stomata and hence prevent transpiration. Resistance, abscisic acid increase the resistance of the plant to the cold and other type of stress. It is therefore also known as the stress hormone. Starch hydrolysis, abscisic acid inhibit gibberellin mediated amylase formation during the germination of the cereal grains. Flowering in small quantity, abscisic acid is known to promote flowering in some short day plants. For example, strawberry, black currant. Okay, in this the flowering is induced by abscisic acid, that is exception. Okay, parthenocarpy, abscisic acid has been found to induce parthenocarpic development in rose. Now rooting of the stem cutting in the is promoted in some cases by abscisic acid. For example, in case of bean, ivy, poinsettia, that is euphorbia. Okay, then membrane potential, abscisic acid induce the positive surface potential on the cell membrane. Controlled growth, it is antagonistic to the gibberellin and counteract the effect of other growth promoting a hormone, that is auxins and the cytokinin, and therefore keep their activity under check. By controlling growth, abscisic acid play an important role in seed development and seed maturation. Normally it inhibits the seed germination, growth of exercised embryo, growth of the duckweeds and other plants. Next we have is the uses of the abscisic acid. Now anti-transpirant, application of minute quantity of abscisic acid to the leaf shall reduce the transpiration to great extent through the partial closure of the stomata. It conserves the water, that is the reduce the requirement of irrigation. Photosynthesis is reduced to the lesser extent, that is transpiration, that is 56% photosynthesis to the 14%. Flowering, it is useful in introducing flowering in some short day plant kept under unfavorable photo periods. Rooting, use of abscisic acid promote rooting in some stem cuttings. Dormancy, abscisic acid can be used in prolonging the dormancy of the birds, storage organs and the seeds. Here you can see some differences between the abscisic acid and the gibberellic acid and cytokinin. Now let's discuss the interaction among the growth regulators. Growth, differentiation and development process of the plant are found to be regulated by two or more phytohormones acting synergistically or antagonistically. Cell division is promoted by both auxin and cytokinin acting synergistically. The cell growth is controlled by the auxin, gibberellin and cytokinin. Morphogenesis is an interplay of auxin and cytokinin, where both must be present in some minimum concentration. When auxin is in excess, the root differentiation on the callus, while the bud develops in the presence of excess cytokinin. Auxin cause apical dominance. Auxin cause the apical dominance while cytokinin overcome the same. Auxin, cytokinin and ethylene produce male flower. Male flower is produced by gibberellin and auxin, okay, and ethylene. Cytokinin and auxin prevent senescence while it is stimulated by the abscisic acid and ethylene. Abscisic acid antagonize the effect of auxin, gibberellin and cytokinin to the growth. It promotes flowering in some short day plants while the gibberellin do so in the long day plant. Cytokinin cause stomata to open while abscisic acid results in their closure. Dormancy of the bud and seed is mostly due to abscisic acid. The same is broken by the gibberellin. Cambial activity and fruit growth seem to be required auxin, gibberellin, and cytokinin. Abscisic acid check the C.